My name is Laura Reich, and I am presenting on my research entitled Utilizing Behavioral and Molecular Techniques to Study Gap Junction Channels. Now, you might be wondering, what are gap junction channels and why do we care about studying them? Well, with that question in mind, I went to back up and widen our lens a little bit. First and foremost, animals exhibit motor behaviors, and the nervous system and musculature coordinate their activity in order for these motor behaviors to occur. Furthermore, both the nervous and muscular systems utilize gap junction channels in order to communicate. Gap junction channels allow for the direct flow of small molecules between adjacent cells, and they are composed of proteins known as connexins. The connexin family is large and complex, and we don't know how individual connexins contribute to behaviors. So this leads us to the big question of my research project: how do individual connexins contribute to behaviors? Understanding this can help us to better understand ways in which the nervous and muscular systems coordinate activity. Embryonic zebrafish serve as a great model organism to tackle this question because they elicit a behavior that is known to require gap junction channels. This behavior is spontaneous coiling, and it occurs at 17 hours post-fertilization and persists until about 24 hours post-fertilization. The coiling circuit is what drives this behavior and contains those gap junction channels. However, we do not know the specific connexins that comprise these gap junction channels. Thus, we can use spontaneous coiling as a proxy to determine the individual connexins in the coiling circuit. As a first step at this, we decided to develop an automated behavioral tracking system. Automated because manual tracking is inefficient, time-consuming, and error-prone. And this system can help us screen for coiling defects and connexin mutants. If we find a connexin mutant that has a coiling defect, that would indicate to us that that specific connexin is required in the circuit. And to validate the accuracy of our system, we decided to analyze coiling behavior in wild-type and Connexin 46.8 mutant zebrafish. This connexin is already known to be expressed in the slow muscle fibers of the coiling circuit at 1 day post-fertilization, and these mutants have a clear coiling defect we can analyze to validate our system. We use DeepLabCut or DLC to do this. It's a neural network that can be trained to track specific points on an organism as it executes a behavior. To train a DLC network, we first captured 150 frames of wild-type coiling behavior. Then, we manually defined 6 points on the frames we wanted to be tracked. Then, after that, we trained the DLC network using the UO to Lapis supercomputer. After training, you should be able to provide DLC with a video of coiling it hasn't seen before, and DLC should be able to place those 6 points on the organism and track their placement over time. And we gave our DLC network novel videos and found that yes, they were able to be tracked. The images on the bottom middle of my poster show trajectory plots for a wild-type and Connexin 46.8 mutant. Each trajectory plot shows the movement of the 6 points over the course of a 30-second video. As you can see, the wild-type fish has a large range of motion in the middle of tail and end of tail points, and this range increases over development. This is not the case for the mutant. Thus, through the DLC data alone, we can see and confirm defects in the Connexin 46.8 mutant coiling behavior. In the top right, I have box and whisker plots I generated that also indicate that Connexin 46.8 mutants have smaller tail movements across development, just by focusing on the x-coordinate of the end of tail point. So, we can validate the accuracy of our system just using these mutants. The next step for my research project was to begin to determine what other connexins are within the coiling circuit. We performed antibody staining in order to see if Connexin 36 fish orthologs localized to coiling circuit neurons at 1 day post-fertilization. And if you look at the antibody staining, you can see by the white arrowheads areas of punctus staining of the Connexin 36 marker right at the cluster of cells where coiling circuit neurons are located. This suggests to us that the Connexin 36 orthologs are forming gap junction channels at coiling circuit neurons. In terms of future directions, we can first optimize the DLC network to ensure that it is properly tracking every point. Then we can perform another round of antibody staining, as well as RNA in situ hybridization, to confirm that the neural connexins we noted in our antibody staining are actually at these coiling circuit neurons. And then lastly, we can use homozygous mutants for those 4 connexins and run it through our DLC network in order to see if those connexins are required for spontaneous coiling. Thank you so much for listening.